Part 2 is microscopy, so it's about microscopes and looking at and observing cells with a light microscope. So here we have a light microscope. You might be expected in an exam to be able to label these things. So you've got the eyepiece, you've got the objective lenses that is not actually shown in this diagram, unfortunately. Objective lenses are the ones that you can rotate and they increase the magnification. You've got the stage, you've got stage clips, you've got the microscope slide on the stage, you've got the light, so the light passes up through the lenses, through the eyepiece into your eye, and then you've got a focusing wheel, so it's either a fine focusing or coarse focusing wheel. So microscopes, they use lenses to magnify or to make bigger and increase the resolution of images. So resolution of a microscope is how well it distinguishes between two points that are close together. So it's the degree of detail. The best thing I can describe is if you look on your mobile phone camera, you'll notice that if you try to look at things at great distances, that it might lose its resolution. It depends how good your camera is. So cameras are good at magnifying, but better cameras are really good at giving good resolution. So microscopes are used to view samples that may not be visible to the naked eye, and they allow us to resolve detail that cannot be seen by the naked eye. So let's have a look at the left hand side. So this is about magnification. So here's an actual size of this blue dot, okay, and you magnify it, and that's called high magnification. So when you magnify things, the image is much bigger than the actual size, okay. Whereas if you have low magnification, the image is still magnified, but it's several times bigger than the actual size. If we look at resolution on the right hand side, so if you can see, if you've got high resolution, the image is clearer and it is more detailed, whereas a low resolution, it's less clear, less detailed, and it tends to be blurry. So you will need to know about two types of microscopes. So on the left we have the light microscope, which you've probably used in the lab. So we know that light passes through the specimen. You can view living cells or organisms, which is an advantage, but they do have generally lower magnification and lower resolution than electron microscopes. Now electron microscopes, they don't use light, they use electrons. So electrons have to pass through a tube. You can see the tube there. It's in a vacuum, and that is why specimens are dead, because there's no oxygen in the vacuum, and they give much higher magnification and much higher resolution than microscopes, light microscopes. You'll need to know about the advantages and disadvantages of both. So an advantage of a light microscope is they can be used to study living cells. They are very cheap and they're portable, but they have lower magnification and lower resolution. Whereas electron microscopes, they have higher magnification, higher resolution. However, you cannot study living cells due to the no oxygen and high pressure in the tube. They're very expensive, they're not portable, and also you need a lot of training and a lot of skill to use one, whereas in a light microscope you can learn the skills very easily. So let's talk a bit about scale and size. So you will need to know a bit about scale and size. So as it says here, the human eye is possible to see a large cell with the naked human eye. So that's what we call 50 to 100 micrometers. The light microscope has a maximum resolution of what we call 200 nanometers, and the max magnification is times 2000. And you can study large parts of cells, organelles, such as the nucleus. The electron microscope has a maximum resolution of 50 picometers, so that's times 10 million. So you can study really small subcellular structures such as ribosomes and plasmid DNA, strands of DNA, chromosomes and things like that. So you will be expected to do some calculations. So what I've got here is what I call the I am triangle. So this is a very good way to remember how to do the different equations. So for example, if you wanted to find the actual size of an image, what you would do is you would measure the image size and divide it by the magnification. Some useful things here, so conversions, so micrometers into millimeters, you can divide by 1,000. If you want to convert millimeters into micrometers, you times it by 1,000. And also you'll need to know a bit about standard form. So what I'm going to do now is just to go through some examples. It's best to use some examples. So here we go with the exam checkpoint. So question checkpoint. So again, what I would like you to do is to pause this and see if you can answer this question. So pause now. So a student used a microscope to observe an animal cell, and it shows the student's drawing, and you can see they've measured it in millimeters, so the length of it, and it says the real length of the cell was 280 micrometers. And it says calculate the magnification of the drawing. So if we go back to this, so magnification is the image size divided by the actual size. So in this case the image size is 112 millimeters and the actual size is 280 micrometers. So what you're going to have to do is convert either the micrometers into millimeters or the millimeters into micrometers. Okay, and what you do is this. So the 112, you can convert it into micrometers, so times it by 1,000, or you could take the micrometers of the real length and divide it by 1,000 to get it into millimeters. So you have to make sure you've got the correct units first. I probably would convert it all into micrometers. That's what I always do with my class. So I always say if you measure in millimeters, to get micrometers, you times it by 1,000. Okay, so if you do that, so let's look at the 112,000 divided by 280. So that gives us times 400. So that would be the correct answer. Let's have a look at another one. Okay, so again, the same as before, I'd like you to have a really good look at this. I'd like you to pause and have a go at this question yourself. So 3, 2, 1, pause. Right, so the image shows some muscle cells from the wall of the stomach as seen through a light microscope. And it gives you what we call a scale bar. So that scale bar is 0.1 millimeters. And it says the scale bar represents 0.1 millimeters. Use a ruler to measure the length of the scale bar. So we'll measure the length of the scale bar with our ruler and calculate the magnification. So what you've got to do is basically measure that distance. So that distance, I measured it as 40 millimeters. So what you need to do then is convert it. Well, you don't need to convert it actually. So you can then just divide 40 with 0.1 and that will give you times 400. So you don't even have to measure the cell.